Hi, everyone. Just want to take a moment to thank Mason and the community for the introduction and organizing this event. Let's get right to it. So I'm going to talk, my introduction mentioned cotton. We're going to be talking a little bit more about maize. So this is the outline for the rotation project that I did with the Murray Lab as the beginning of my Ph.D. student career. So we're going to talk a little bit about how deep learning-based high-throughput detection of flowered maize plots is a worthwhile venture to replace traditional phenotyping approaches. And we'll go through kind of the ins and outs of what computer vision is. This is about a year old in computer vision as far as the models that you would employ. It's been growing rapidly, so multi-layer perceptrons, adversarial neural networks, autoencoders, all of these technologies are things that I would definitely want to pursue, but I would like to consider this as kind of computer vision 101. This is the introductory to this process. So for maize specifically, we're going to be talking a lot about tassels and in general the flowering window. And for that information, we can capture it with drone or UAS systems, and the rate at which those images are captured outpaces analysis. We get, in one flight, you're looking at a field with 500 plots, and we can do that in a very short period of time compared to sending people out to that field, so it simply just outpaces manual observations. It reduces the labor-intensive manual approach, which can be not just labor-intensive, but relatively subjective. And one of the major components to the lack of adoption of these technologies is not just the cost of the devices themselves, but the cost of the software and the training and the understanding of the analysis and the utilization of that software. And this was a fairly recent review that showed almost half of breeding programs are reluctant to employ these things because of those complex barriers. So the question is simple. Can drone-based tassel detection via a CNN model effectively replace manual methods? Will we get higher repeatability? Can we lower our labor, cost, time input, and get something more meaningful from it? So the important aspects of this data set is we've got two different parents that are used at several different locations. In College Station and Arlington, we have the same. In Madison, we have a different one. In our 5 fields, the 3 fields in Texas each had different management conditions, whether they were optimally planting as far as date, whether they were irrigated or dry land. And then we've got 2 other optimal locations. We began processing these flights. So we fly the drone over the field. We begin by cropping each individual plot from the flight where we see 0% tassels. And then the last flight is where all of those plants have tassels. So the idea of the emergence of those tassels being observed is very clearly present within them, and that comes from 15,000 cropped row images. So DTT and DTA, these are the phenotypes that we're talking about. DTT is days to tassel detection, right? We fly a drone, we obtain the RBG image. Within that field, there are going to be some plots that have no tassels, some plots that do. We crop those and label them as a 0 or a 1. So how many of you would like to go and label 15,000 cropped images? Probably not many of you, right? This is a process that, because we've done it, we can use this data set to then advance it to other images that haven't been labeled, and we're going to talk about that later. And then we're comparing that to the manual DTA, where we send workers to the field, we observe the presence or absence of a tassel and record the date for it. The uncanny valley nature of this picture is because I asked ChatGPT to make an image of Texas A&M students in a cornfield looking at maize tassels, and that's what it thinks we look like. So what is a CNN? For those of you who don't have familiarity with computer vision, this is how a computer interprets an image. For this image, it's a 26 by 26 pixel image, so if you were to count the cells, it's 26 by 26. For those of us from Texas A&M, very familiar with this letter. And the numbers represent the intensity of those pixel values. So we can see visibly the red is more intense at a 1 than at a 0.7. And the computer is going to go in a reading frame through the image and look at the value of all those pixels. And for us, let's look at the first kind of informative frame, that blue one. So if I put this image into my computer, the only thing that it's going to understand are the values of the individual pixels. Within our computer, we apply a filter. This is a part of the convolutional neural network. And for the most part, these are generated within your software. But if I'm going to basically purposefully make these filters, I could make a left-right side detecting filter. And this is in the computer. So this is just a part of the program. It's what it is. We feed it a variety of different random images. And it reads through all of those 3 by 3, and you can determine 5 by 5, however you want, pixel matrices, and does a convolving step. This convolving is simply just matrix multiplication. We are taking the sum product of those two matrices, so the one within our program and the one from the image, gives us a value, and now we've collapsed that matrix down to its singular value. And if we do that in all of the possible frames, we get to see the sides of that image. Change the filter, change the image. Overlay the images. This is how our computer can interpret that image. I can go and change that input image, and its features will be extracted in a very similar way. This is the most simple application of a neural network. Within your neural network, this is something that we've heard about a little bit already, but hyperparameters are the different components within that architecture that are going to affect the way that we process the image. The number of convolutional layers refers to how many times we reduce that image down in size. We've got the kernel size, the number of filters. All of those are the elements of how the computer scans the individual pixels. And I'm happy to talk more about this, but the major conclusion is that these hyperparameter importances aren't static. Not only cross-crop, but cross-phenotype and cross-model, there are differences where the same hyperparameter may be more important elsewhere. For our model specifically, the number of convolutional layers was the most important, and we found that to be 5. So we reduced the image down 5 times in those convolving steps, and that gave us the highest accuracy. So Optuna, this is a package that we ran on the high-performance research clusters, and it uses FANOVA to determine which setting in those hyperparameters gives you the best accuracy. I'm happy to talk more about that later. So where we can apply this, if I look at all of the DTA and DTT value from most of our locations, these are going to be all the ones that use the same parent, what we can see is a fairly close trend. It's definitely not improved. The repeatability is slightly lower, the correlation is slightly higher, and the error is definitely comparable. But there's something special about one of our locations, and we're going to talk about what makes that location special in a little bit. If we look at just Madison, Wisconsin, we see a different trend where the repeatability, the amount of genetic variance potentially captured in this approach, there is more of that in the DTT approach than in the DTA approach. So not only is the manual method going to be more labor intense, it's going to cost you more, it is potentially less informative than a deep learning based approach. What we did to determine exactly the optimal accuracy for this was to perform the standard training and testing split of 80% of Texas in 2020. So all of the College Station, Texas 2020 flights, we take 80% of those and we divide it into the 80-20 train test split. So the computer vision model is only looking at a subset of the images in Texas from 2020. From that, it is able to determine accuracy of the unseen image, first from the 20% that were not seen in Texas 2020, and then we're going to go on and apply this to the unrelated genotypes and the other unseen environments. One of the noteworthy elements of this is, you can run these epochs forever, but early stopping is one of the most effective methods to limit overfitting, as well as kind of just limiting computational resources. You can stop it at 20 instead of running it to 50 because we see the overfitting begins to extend and accuracy kind of levels out. So this is my favorite figure from this. This shows that deep learning enables scalable, high throughput flowered maize plot detection. This is a plot with tassels. This is a plot without. And we can see that after we've reduced down to layer 6, there is a difference in the activation, which shows that the computer vision model definitely understands what it's looking at. The main reasons why we have some differences, we're going to see these ground sampling distance values are different. This is largely determined by how high you're flying your drone. The higher the drone, the faster it is, the lower the resolution. So there's a trade-off. And if we look in Texas 2021, we have a very high value, which is a very low resolution. How did that affect us? Well, resolution and accuracy seems to have a very clear pattern. Our lowest resolution When the actual class, that means we're looking at an image with no tassels. When it's a low resolution, it is more likely to be labeled as having tassels than at a high resolution. So at our highest resolutions, we're looking at incredibly high accuracy in those labels, whereas for the low resolution, we're getting a little bit more of that mistake here. The other main difference is going to be flight density. So if we look at Texas, we're getting flights that don't occur as densely as in Wisconsin. These are very dense that occur within that tassel emergence period, which means we're truly going to capture the day that it happens. The issue comes when if we have a flight that occurs on day 71, and those tassels emerge on day 72, 73, 74, if our next flight is on 78, those will all be recorded as day 78. And that affects our correlation by making these kind of flat lines here where we're restricted in the amount of DTT values that are present. So they all kind of fall in that one. Even with this, we still have a high enough correlation that retroactively, this is stuff that we did in 2024, this could still be applied and work on 2020 and 2021. So it's highly correlated, and in general, what I believe are the most important facets of this is that automated phenotyping reduces labor. That much was very clear. The deep learning can enhance selection quality and consistency. We saw higher repeatability for DTT than DTA in Madison. It lowers the barrier to AI adoption. This is open source. Everything is available. Everyone should have access to it. Future work should pursue a variety of different sensors. And I want to just thank everyone for their work in contributing to this. Thank you so much.
Hello, Nick.
Cody.
What is the cost difference? You mentioned the limitation of your flight dates. Is it more cost effective to fly the field every day, or is it more cost effective to have one person go out and look at the tassels in person every day?
Well, so as far as that specific cost, there's a very clear 7 to 10x fold decrease in the number of hours. So right away, you're going to spend fewer hours pursuing this technique. And that 7 to 10x fold includes learning Python, learning Keras and TensorFlow, learning how to code your first neural network. All of this was the time from start to finish where it included all of the training that I did myself. So anyone with no experience, you could expect a 7 to 10. If you have coding experience, it could be even greater than that. And as far as flying multiple times per day, it would depend on the phenotype of interest. So with Maize tasseling, we wouldn't see enough difference within a day, but between days, certainly. If you have 10 flights, if your budget accounts for 10 flights, it may be best for your phenotype to do 5 flights, 2 days back to back, 10 weeks, 1 flight a week, or 1 flight a day for 10 days. It's going to depend on the phenotype.
Any question?
What about the computational needs? You mentioned big high-performance cluster. Do you need to have that to be running on a PC or what?
So the appeal of my model is that the model itself can run on a PC, but it was optimized on a standard either high efficiency laptop or desktop with a good enough RAM that would be deemed modern. Definitely not an old machine could run it. But the optimization was done on the high performance research clusters. So we used that to just test every single possibility of hyperparameter, and that is an intensive process. But because we've done it, and because our data set, it's 15,000 images, it's a variety of different orientations and pixel values, this model, because it's been optimized, should function on other cropped Maize plots.
Thanks to our speaker, Shepard. Thank you so much.
Thank you.